pioneer is Wenting Zhang. She's currently a PhD student in RA at Stevens Institute of Technology. Working in materials science and engineering for years, Zhang's award-winning and patent-pending research is based on an ex vivo method to determine effectiveness of personalized treatment for multiple melanoma and to model the bone metastasis site of prostate cancer. She's going to be speaking on this topic tonight. Please welcome Wenting Zhang.
Thank you, everybody. I'm glad to present here our exciting new work and share our new idea with you. My topic today is patient-specific model for multiple myeloma. I would like to first introduce our team to you. As a collaborative project, we have people from different areas for sure. We have professors from material science, chemical engineering, medical engineering, and chemistry, biology department from Stevens. And we also have Dr. David Siegel, who is one of the leading multiple myeloma clinician from Hackensack University Medical Center, and also some other research scientists as well, which are very helpful. And so our project currently is supported by an NIH funding. So to begin with, in the past 6 years, we know that there are many diseases have been making great progress in the disease control and for finding a cure. But for cancer, it's not like other diseases that is still making very limited progress. That means still almost no cure. And it's causing a huge burden to the society and to the families. So we all know that there's no cure, but why? So based on our understanding, a major reason is that cancer is not one disease, but many. Even for the same type of cancer, like lung cancer, different patient has different cancer cells. If we apply the same drug to all these lung cancer patients, they will have different response. So regarding today's, that's how people came up with the idea of personalized medicine. So people has developed this personalized medicine in the past 10 decades only after the human genomic plan has finished. And we're going to address some of these plans later. But one major step in the personalized medicine is that we have to build up a patient-specific model. That is for applying different drug combination and regimens to those patient-specific models, and then we can read out different drug efficacy for specific patient. So the multiple myeloma is a disease model we're going to talk about today, and it's a very good example for personalized medicine, and I will tell you why. Multiple myeloma is a plasma cell cancer which grows in bone marrow, and it's the second most blood cancer in U.S. And unfortunately, it's still no cure. Even for the early stage multiple myeloma patient, the only treatment strategy right now is just to control the disease progression and prolong the patient's life. With an advanced treatment right now, the patient can survive about 5 to 7 years. So the difficulty for treating multiple myeloma is that no matter what kind of drug a patient take, they will eventually get relapsed. It's only as a matter of time how long they get relapsed. So that came up with the question that how can we choose the right drug for the patient to prolong this kind of period. So that's why the clinician doctors are very interested in developing a multiple myeloma personalized medicine. So regarding to multiple myeloma personalized medicine, the challenge is that we have to figure out the dilemma for multiple myeloma, because this disease, they cannot be killed inside of the body, but once they are coming out of the body, they will die instantly within 3 days. So that's not good for drug test. So we have to figure out how to maintain and expand multiple myeloma outside the body. So right now, there are mainly 2 ways to maintain primary human cell outside the body. The first one is to culture cells in a petri dish, but as I said, it's not going to happen for multiple myeloma. So the widely used ex vivo model, that is to culture human multiple myeloma cell outside their body, is to use a mouse model. That is to we take patient sample out of the human body and then inject them into a immunodeficient mouse, and then we test. We can know a lot of multiple myeloma biology from this, but when it comes to the drug test, this mouse model fail more often than not. So we have to find a better tool. So that we came up with the idea that to use a human tissue engineering on a chip. That is, we have to use primary human cells, and we have to reconstruct tissue microenvironment, which include the primary cells interactions and the nutrition, growth factors, and all the mechanical force. So we know we cannot provide or reconstruct the whole human body, but the snapshot we are reconstructing for the phenotypic, the tumor cells, and this will give us a snapshot of the drug test, and this is good enough for the personalized medicine. So we know that multiple myeloma will grow in the bone marrow, so we are trying to reconstruct every element in the bone marrow microenvironment. That is to include all the bone marrow cells and the bone cells and the growth factors, everything. So what we do here is we first reconstruct the bone structure in a microfluidic device, which include the dynamic environment. And then we get patient bone marrow biopsy and label them in order to distinguish them from the bone cells. And then we can culture the bone marrow cells directly onto the pre-made bone tissue, and we succeeded. So this technology has been patented and published on a journal paper, and we have won New Jersey Hall of Fame, Inventor Hall of Fame. So this is a picture showing that the red cells are bone marrow cells, mostly multiple myeloma cells. They are growing happily in the pre-made bone cells, which is the green cells. So this is data showing that after 21 days, the multiple myeloma cells are still growing, expanding in our in vitro model. So we test 7 days and 21 days for the expansion, and we use CFSE and the flow cytometric technique to test the expansion. And we tested that they can expand up to 5 times. So this is a good proof that we can really expand them. And this is a video. Unfortunately, it cannot play today. So we've take some time to find out that osteoblasts, that is the bone cell, that is a very critical element for this in vitro model to maintain multiple myeloma cell ex vivo. And this is another proof that they really attach to osteoblast. So this is how we plan to do the personalized medicine. We basically take the patient's bone marrow cell out of the body, and based on the pre-selected drug, we test it on the tissue sample, and the doctor will read based on the readout and select the most effective drug. So this is our. Thank you so much, I'm sorry.
I'd now like to open it up to the panel.
So it seems like we didn't get all the way there, but do you have data? Because if we culture the cell, is it likely the same tumor? I don't understand exactly that. And then what's the data been in the drug cocktails? Because I'm assuming that there's multiple parties that would be involved in allowing those drugs to be administered. Is there any other issue there potentially?
Yeah. That's a good question. That is the question we're going to address next. Because firstly, we have to conquer the difficulty to expand and maintain the multiple myeloma cell ex vivo. If we can do this, that is a proof that we are recapitulate the in vivo environment. That is, we are recapitulating all the critical environment for them to survive. So, so our next step is to test by drug test. So we have to compare parallel to use the same drug to test the patient ex vivo model on our model, and then compare this if they have the same effect as the patient take in vivo. So doctors will guide mostly on this comparison. So we are focusing on the this model reconstruction.
Right. Would that be the way to market, to go to the doctors and get them to prove for standard of care? Just to understand how we would actually get it there.
Yes, basically yes.
Okay.
Yeah. So doctor will tell us how to make this very clinically relevant.
What are some other challenges that you foresee in bringing this to market, and how do you plan on addressing them?
I think the difficulty is that it takes time for us to validate this model, because multiple myeloma it's a chronic disease. It takes time for the disease to progress. So we want to see from different time point if we take a patient sample out of the body at time 1 and test them, and then after some kind of period we take them out again, maybe the phenotype will change. So because the cells are in a different environment, so I think we have to take we have to know how long this phenotype can be kept in our the model.
And talk to us a little bit about, I know multiple myeloma and how common it is, but talk to us how this process can be applied to other chronic cancers that people are dealing with. Is this, you think, applicable across a broad spectrum of cancer patients, or just really specific to multiple myeloma?
That actually I'll address in the last slide. So not only for multiple myeloma, but we also want to broaden this model to other bone-related cancer disease, because like breast cancer and prostate cancer, they are very likely to have bone metastasis site. So there are some drugs addressing the breast cancer and prostate cancer in their bone metastasis effect. So we can test those kind of drug on this broadened tissue model.
Great. We're a team. That's why I wanted to make sure you got a chance to say that. So thank you again for coming tonight. We appreciate it.
Okay, thank you.